biology. Hope you're doing well when you get this video. Wanted to walk you through the Catalase Lab report. So here is Catalase. Multiple colors indicate multiple subunits making up. This is the quaternary structure. Catalase is an enzyme that breaks down this reaction right here. So it takes hydrogen peroxide, that would be the reactant, and it breaks it down into water and oxygen gas. So it happens in living tissue, happens in you. So I'm sure you remember our experiment of a couple of days ago. So this is the reaction that it catalyzes. Again, speeds up very quickly. So hydrogen peroxide, why is this important? Because it's a dangerous substance. It happens from metabolism normally, but it has to be converted into less dangerous substances. So catalase is used to quickly do this. So here is a diagram of exactly, a very basic diagram of exactly what catalase does. So here's your enzyme substrate complex. So we take hydrogen peroxide, which is a substrate, binds to the active site right here, and then the enzyme has an induced fit, kind of like the lock and key example, and it breaks it down, catalyzes the reaction into water and oxygen, which of course is not dangerous to our bodies. So what can we do though to change the enzyme? So this lab was all about what affects enzyme activity. What affects it? Okay, and so we tested out temperature. And one was boiled, which was 100 degrees Celsius. One was at room temp, and then one was cold. I forgot, exactly what each class was, but one was like 5 degrees or 6 degrees Celsius, very cold. Okay, and so we investigated that, what affects enzymes, and we investigated temperature. So this was our setup. I'm sure you probably remember it, the floating disk assay, which we just kind of drop it down, and then inside the disk it floats because there's bubbles being made if the enzyme is working. If the enzyme is working, oxygen is being made, and so there you go. And so you had your control, and then you maybe had like hot, room temp, ice, and then we did multiple experiments. So what exactly am I looking for the lab report? The lab report itself, and this is not AP. So you have your introduction, your basic description of how enzymes work. So this is a paragraph. Okay, and so basic description of how enzymes work, brief summary of why catalase is important. And then, of course, always want to understand the why, right? Why did we do this? Why, what was the purpose of this investigation? And then, yes, definitely write your hypothesis, if then statement, okay? I do want you to draw your experiment, which is basically up here. You can just draw it. Drawing helps us understand exactly kind of what we've done. And then a table and a correct graph, okay? So just for your group's data. So I do want two forms of visual representation. I want a data table, and then I want a correct graph correctly illustrating what you saw in the lab, okay? So finally, the conclusion is, was your hypothesis supported or rejected? Again, hypotheses are not necessarily right or wrong. They're supported or rejected in science. And why? Why do you know this? Use data to show this, okay? What did temperature do to the enzyme's activity? Again, use data there as well. And then why do you think that is? Your conclusion should be different than your lab partners. Everybody's writing this up, okay? And then finally, yes, of course, everything that we do involves human error, especially in high school biology. So what possibly could have happened that would have made this experiment faulty, or some areas that we might need to adjust if we were to run it again. Again, repetition is beneficial in the realm of science. So again, this lab report should be written in your lab folder, your journal, okay? Again, at the end of the year, we want to see everything that we've done, so write it in your journal, handwritten. If you want to type it and print it and staple it in there, that's fine, but I do need it in your lab journal, okay? If you have any questions, please email me. Again, I hope you're doing well.